I'll introduce today Richard Colwell. He's an NSF graduate research fellow pursuing PhD in material science and engineering. And he works in the Eleftheria Roumeli's group, and he explores biomass-derived materials for sustainability applications. He has also worked at several different places, at Amazon Worldwide Sustainability and Tender Food, and he did his undergrad in MIT. And also he's a Colwell fellow.
Yes.
So we're happy to have him today. And he will speak about predictive modeling of Whole-Biomatter algae bioplastics.
Awesome. Thank you.
Thank you.
All right. I will go ahead and get started. So the one thing I want to first just preface my presentation with is, as was already mentioned, I'm in MSc, not in computer science. So this is kind of a side project of mine. So you might know the data science and the actual ML better than me. I'm going to do my best. But really, this presentation is all about applying existing tools to an underexplored material system. So briefly, I'll talk about the issue with plastics, which I think people are pretty familiar with. I'll talk a little bit about what our lab actually does, some background on material science for those of you who maybe are not familiar, and then I'll share a little bit about the data sets I worked with, talk about some of the methods, get into results, and then kind of some of the next steps for this project. So I won't spend too much time here. I think most people are pretty familiar with the plastics problem, but just some high-level statistics. In 2019, we were producing almost 500 million tons of plastic globally. That has only gone up in the past half decade. And about half of that goes to landfill, and about a quarter basically ends up in the environment, either in uncontrolled dump sites or in the oceans. And this use and production and waste from plastic is expected to only continue to grow, probably by about 70% by 2040. And so our lab has a solution to try to replace some of these plastics, and we call these Whole-Biomatter bioplastics. And it sounds pretty complicated, but it's actually pretty simple. So we use thermomechanical processing, which is really just a fancy way of saying we apply heat and pressure to simple biological powders. And we are able to make plastic-like materials from all sorts of different feedstocks. So we've done matcha, we've done dragon fruit powder, and then spirulina is a particular algae we'll talk about today. And these plastics have pretty similar performance to incumbent petroleum-derived plastics, but the one key difference is they won't last for decades or centuries. And if you throw this outside a year from now with the Seattle rain, I promise you, you will not be able to find it. So why do we actually care about algae? There are a couple of key reasons. Compared to a lot of sustainable plastics that might depend on corn or sugarcane, algae does not necessarily compete with land for food crops. It's fast growing, rich compositionally, meaning there are lots of different types of algae that might have different compounds in them that gives us different properties, which I, as a material scientist, like the idea of being able to exploit. And lastly, algae is able to sequester a lot of carbon, so it can be a carbon negative material. So now to get a little bit into mechanical testing, just a very quick crash course. The type of testing we do is called three-point bend. And the reason for that, if you look in this diagram here, is basically I have a sample across, maybe I should have made this bigger, but across two supports, and then there's another point on top that is going to come down and push on my material and basically bend it until it breaks. And so this figure in the middle is what we call a stress strain curve. And essentially what this is measuring is how much stress, which is just normalized force, how much force a material is experiencing as it's bending. And so we have a couple key properties that we care about. I'll go top to bottom. So this top one, sigma F, that is our flexural strength. That's basically how much stress or how much normalized force can I apply before the material breaks. Then coming down here, we have the flexural modulus, or we would colloquially call that the stiffness. And so that's just the slope of our triangle. We have the area of our triangle is the toughness. It's how much energy a material can displace during deformation. And then last is the strain to break. And that's basically just a measure of how far did I bend it before it snapped in half. And so for three-point bend, it kind of gets complicated with the equations. So these aren't really important. It's just important to know we're normalizing these properties based on our samples. If I have a huge sample, it's going to take more force to break it. And so this just makes sure that we're measuring a material property and not a property of a single beam. And lastly, in the introduction, I'll talk a little bit about why these materials can be hard to deal with. And one big reason is there's a lot of experimental noise coming from a lot of different sources. I mentioned that we make these samples by heating them under pressure, but you can imagine, even if I think it's set to 140 Celsius, if it's a cold day and the room is a little chilly, basically that heating system is going to be fighting to keep in thermal equilibrium. If it's a really hot day, maybe the gradients are slightly different. So it's not going to be one single temperature throughout the whole system perfectly. Similarly, the pressure doesn't stay magically the same. Two people might fill our mold slightly differently, so the powders are going to be slightly uneven. That can actually cause subtle differences in what we measure. One that I'm currently studying more thoroughly in my PhD is the effect of water. Water is everywhere. We can't see it in the air, but especially in Seattle winters when it's very wet, it actually can affect the results of our experiments if we're not very, very careful. Similarly, if I'm measuring my samples, there's a little bit of uncertainty in all of my different measurements. And so these all can propagate to mean we don't get a single consistent measurement every single time we make and test a sample. On top of that, you can imagine if I grow some algae this year and I harvest it, that's going to have a whole soup of different biomolecules, so different proteins and carbohydrates and fats. It sounds a lot like a nutrition label. That's kind of the way I think about it. But from year to year, if it's hotter or if maybe there's more salt in a particular part of the ocean, that might mean that there are more proteins or less proteins or different types of proteins. And so feedstock variability year to year can also change. And the last one, and probably the biggest motivation for this work today, is these samples actually take a considerable amount of time to both fabricate and then to test. And this means that we can't just generate 1,000 samples of something and run a machine learning model or 10,000 samples. We're typically making much smaller amounts on the order of maybe 100 for a paper that took years to produce. And so the big question that I wanted to ask with this project is, can we build models to map the processing and composition of these materials to the properties under the constraint of limited noisy data? And today I'm going to look at 3 data sets, and I want to shout out 2 people in my lab, Harish, who's actually here in the audience in the middle, and then Ian Campbell as well, who actually did the hard work of producing the samples that I'm going to look at today. And I'll jump right into what those data sets are. So the first one was kind of I would say the big paper that demonstrated this as a method coming from our group. And in this paper, Harish took a single feedstock for at least the data that I'll look at, spirulina, which some people will use in like a green protein shake. It's a cyanobacteria that has gone through a lot of taxonomical changes. So if you look it up next year, it might not be called Athrospira platensis as a warning. But essentially what he did is he varied the press temperature and the pressing force and looked at what is the effect of that on the properties. One key call out here is, as we can see, actually you can't see it on, okay, there we go. If we look down here, these hashes are basically demonstrating those samples at low force and low temperatures were actually untestable. They were so weak coming out of the mold that you couldn't even get them out to be able to test them. And that's going to be important later. But the key thing is we change temperature and pressure, we measure our properties, and it's one single composition. So the feedstock is the same. The second data set is the flip of that. And so in this paper, Ian's goal was to look at a more simplified system. So I mentioned algae, like your favorite food products, has a lot of different compounds in it. So we have long carbohydrates, we have different types of proteins, maybe there are salts. And that can make it really hard to understand what's going on when we make these samples, what's affecting the way that they perform. So the idea behind this paper was to say, let's take 4 more known materials that can represent the broad class of biomolecules and just change the ratios of those so that we can better understand what is the role that a long structural carbohydrate plays, what is the role of protein, what are the role of small molecules like small carbohydrates and fatty acids. And basically, we can just change the recipe for our beams or for our materials and see how that affects properties. And so in this case, we kept the pressing conditions the same, so the same temperature and pressure, and we're just looking at different compositions. And then lastly, we're looking at the Hawaiian algae paper that was recently published. And the fact that it's Hawaiian algae is not really the key today. The key is that we have 4 different types of algae. So we have 4 different compositional profiles. And for each of those, we try 4 different pressing conditions. And so that's going to be kind of the main reason why we include this. So study number 1, it's all the same composition, just different processing. Study number 2, all the same processing, different composition. Study number 3 combines both of those factors. So what did I actually do since I didn't make these samples? These aren't my papers. So the essential goal was to look at how we could use simple machine learning models in order to map, like I said, the processing or the composition onto performance. I won't talk too much about linear regression because I think most people probably are familiar with lines. But I will talk a little bit about Gaussian process regression, which is the most flexible model that I investigated. So for people who aren't familiar with Gaussian process regression, like I was not prior to this project, essentially a Gaussian process is going to give us a probability distribution over some family of different functions we can consider. So when we think of linear regression, we're basically saying, assume that the function is going to be linear. And in this case, we have a lot more flexibility over what functions we are allowed to consider. And this is going to have both a mean function, which is giving us the actual predicted value, but a key portion is the kernel choice. And that's going to tell us basically how smooth data points that are a tiny change in the input away will be in the output space. And I think this figure is actually a really great way to visualize that. So on the left, what we see here is these are just some kind of squiggly looking functions. We're going to start, this is a subset. So imagine we might have thousands or maybe infinitely many curves that look kind of like these ones on the left. But then what we're going to do is we're going to actually introduce our experimental data and use that to adjust which functions we actually think might be representative of our data. And so you can see here we have 5 of these x's, and essentially we are going to then upweight the functions that actually might better fit that data. And so what you see here is our confidence interval around our data points is actually getting really close to that ground truth data. But for places where we don't have data, we see our confidence interval is really, really wide. But this is kind of the general idea, is we're going to start with a bunch of functions, and there's some probability that they might be right for our samples. We'll show some experimental data, and then we'll refine what functions we think are actually a good guess. And I guess this is just kind of repeating that. So we start with our family of functions, we look at some experimental data, and we say, what does the distribution need to be to better fit our observations? So I mentioned kernels, and this is actually a pretty important piece here. So looking on the right side first, I want to call it these 4 on the bottom. So we have SE, RQ, ME3, and ME5. So these are all, like I said, they're different kernels. And the important thing about them is that they all make slightly different assumptions about how smooth our data is going to be. And so if we see that one fits better than another, that's actually telling us something about the actual shape of our data. And the left just shows kind of as we move further in our standardized feature space. So for example, if I'm just considering temperature, if I move from one press temperature to another, how similar or how dissimilar should other materials be? And that's basically what these shapes are showing us. And we basically use these functions to build a covariance matrix. That's kind of what's happening under the hood. I'm happy to answer questions to the best of my ability after, but I won't spend too much more time on it. The last methodological thing I want to talk about is how are we actually doing cross-validation? For our cases, we are going to do something that is pretty strict, pretty challenging, but also very important because of the shape of our data. I didn't mention this, but for each of our cases, we produce replicates. And that's because for us, we know that there's noise. So we want to make sure that we're actually evaluating a mean, which we obviously can't do if we only have one sample. But if we actually train a model where it might be able to see multiple samples at one condition and then predict on another sample at that condition, it can pretty easily just memorize what did the other samples at this condition look like. And hopefully if our processing is consistent, that's going to do a really good job of creating a model. It won't generalize very well. So instead, what we do is LOCO, which is leave one condition out or leave one cluster out cross-validation. So for each data set, and I've taken the spirulina as an example here, we're going to leave out one processing composition cluster. So we might say, we can't see that condition at 8035. And then we are going to essentially train the model on all of that data. And then we will test on that one left out condition. And then we're going to repeat this so that we've hidden every single condition once, trained the model, in this case, 24 times, and gotten our estimates versus the ground truth data. And the great thing about this is it will tell us, is the model we're building actually good at generalizing to things the model hasn't seen before? And so this is pretty strict, so it's hard to build a good model for noisy data in this way. But it ensures that we're not fitting something that is literally just memorizing the data from our data set. So now I'm going to actually get into some of the results, although this is kind of methodological as well. So in each of these sets, I guess we can see there's both the darker blue color and the lighter green color. What this represents is the blue being our full data set, and the green is half beam or whole beams only, sorry. And so the way to think about this, it's actually motivated by our issue of not having that many samples. So we're trying to increase our sample size. So if I snap a beam in half, I'm left with two halves of the beams. You might say, okay, those go in the garbage. But you can look at that beam and say, what if I snap that beam in half again? Can I actually 3x the amount of data I'm generating? And it's kind of a nifty way to just, maximize the amount of samples. Maybe you could even break those halves in half and, suddenly you're doubling again. But what I found through my analysis is if we include our full data set, we end up having basically worse model performance, both in terms of the maximum possible performance, but additionally, all of our models do perform significantly worse compared to the green. And from left to right in each set, we have a theoretical R squared, the ICC, which is measuring how much variance comes within one condition versus across all of the conditions. And then this is a little sneak peek of the model performance in the third bar. So that addresses the question of should we include these half beams or not? The answer is no. We basically get a worse model, even though in the first case of only using the whole beams, we're cutting our data by a factor of 3. So we're saying I only am taking a third of my data. The models perform much better if we only look at that data. So the second thing that I wanted to check in on, I mentioned that these beams on the bottom were really, really weak, and so they couldn't even be tested. So functionally, we said strength of 0, stiffness of 0, strain to break of 0, toughness of 0, because we couldn't test them. But the model doesn't know that because I don't actually have any raw data at that condition. And so what I wanted to see is, does that affect our model? Or put another way, if I fabricate or if I create proxy data for these conditions, how does that affect my performance? And so I have a very similar plot to the last one. This darker green on the left is now taking that first change of only looking at whole beams. And now we're comparing what if we create this additional data. And again, the big takeaway here is for all of our metrics, basically from left to right, everything is going up. So our max R squared we could possibly achieve goes up in every single case if we add this additional data. The ICC also goes up, which is very similar to the max R squared, and our model performance also goes up as well. So one last thing, I mentioned the selection of kernels is really important when you're using the Gaussian process regression. And one cool thing about these kernels is you can add them together, you can multiply them, you can do all sorts of fun things. And there's actually a kernel called the white noise kernel. And usually we think of noise as not being super great. Usually like noisy data is not ideal. But there's actually a really interesting finding when you have replicate data when you're not using the noise kernel. And so that's kind of what's visualized here. So on the top plot, this looks very similar to the example I showed. So these green dots are the actual data. And then we see we're getting a pretty good fit that seems to trace the trend that we see in the data pretty well. But an interesting thing is our confidence interval shrinks and basically is just at the mean of the data, which obviously can't be right because it's saying we're 95% sure that none of our data exists in that confidence interval, which is not good. We would like to see that our data is captured within the error of the model. Um, on the other hand, if we look down on the bottom, these curves, they look a lot gnarlier, a lot more noise for sure. But the key thing here is we still get the same predictions out of our model. So the function still more or less looks the same, but we're able to generate a more realistic confidence interval that actually does capture the real data. Okay, so now we'll actually talk about the results. What did we find in the different data sets? Um, and I'll give a warning, it's not perfect for every single model. So again, I mentioned we have four different properties. So those are going to be the strength, stiffness, toughness, and strain to break. And so for each of the data sets, I'll walk through kind of what is a model performance for each property. So on the left, we see the strength for the spirulina. This is our best estimated property. And we see even just a simple linear model does a pretty good job. So if you just say there's some factor I'm multiplying temperature by and some factor I'm multiplying pressure by, we get almost a 0.7 for our R squared. But introducing this Gaussian process regression does actually give us significant improvements to our model's performance. Similar story for the stiffness, although the maximum R squared is not quite as good as for the strength. And then as we move to our other two properties, we see very similarly, we're around 0.5 to 0.6 for our R squared. But interestingly, the Gaussian process regression is actually not gaining that much in terms of model performance. In fact, for the strain to break, so how much I can bend the sample, it actually gets the best performance out of just a simple linear plus interaction model. And so this tells us for some properties, we're able to actually learn things about the property space through the different curvature introduced from the Gaussian process regression. For some properties, we actually can't model them much better than this. So some key takeaways from this first system, we do see global trends in our mechanical property response. So if I increase pressure, generally that increases performance. If I increase temperature, that also generally has a positive impact. But there do seem to be some nonlinear regions where GPR is a helpful method. And as we just saw, strength and stiffness are modeled more accurately than strain to break and toughness. And then, as we talked about at the beginning, there are benefits of reducing our data set to only introduce the whole beams, and then the proxy data also further improves our model accuracy. I'll kind of speed through these because it's very similar shaped graphs. The only thing to remember here is now we're looking at changing the composition of all of our beams, and we're pressing them at the same temperature and pressure. And I have a really unfortunate starting result for my strength, which is that all of the models have a negative R squared. And so essentially what that's telling us is I would be better off to just guess that all the samples perform with the mean strength, and I would actually get a better model. Not great. But on the bright side, we look at our stiffness, and that property does seem to actually be well modeled, honestly, through all of the models, but interestingly, again, better through interactions than the Gaussian process regression. So in this case, we see there actually seems to not be this curvature that's captured in the first system, at least not that is getting trained here. And a little bit more of the same from our first plot. We see basically no models that I tested, at least I should say, are able to capture the toughness or the strain to break in this system, which is really unfortunate, but these are what the results are. And so as we saw, the stiffness of our biomatter analogs can be well modeled with simple linear and linear plus interaction regression models. And it also can be reasonably modeled with the Gaussian process regression. But basically everything else we look at, we should just guess. It's probably going to be the average of our data set, which is also a finding. Negative findings are findings too. And last, I'll close out with the Hawaiian algae system. And so again, the big thing here is we have four different types of algae that are all in the same data set, and we press all of them with four different temperature pressure pairs. And here there's one specific thing I want to call out, and that is the difference, and I realize this might not be super visible here, but in these pairs of bars with the hashes, I tried two different things for this model. So I mentioned there are four different algae types. So one way that we could train our model is we could say, for those of you that are familiar with one-hot encoding, I can say, is this algae A, yes or no? Is this algae B, yes or no? Is this algae C, yes or no? And so that's what we see is listed as categorical. So that's the left or the non-hash bars. Um, but we also did run tests to actually figure out how much protein is in algae A, how much protein is in algae B, how much ash is in algae A, which is just if you burn your sample, what's left at the end. So like salt won't burn away. Um, and a big finding here that I think is pretty obvious with this data is the categorical or the one-hot encoding case is able to actually do really, really well, at least if we're looking on the left side for the strength of our materials. As soon as we try to say, can I learn something by just looking at the amount of protein from sample A to sample B, suddenly these models all break down. So they're not able to generalize, at least again for this data set, just based on composition alone. Um, we look on the right side, and I think my big takeaway is, yeah, again, these models are just not cutting it for predicting that particular property. Um, and then on the next slide, we'll have a little bit of a flip of that. So we see strain to break, once again, poorly modeled. But interestingly, this time, the toughness actually really easy to represent with the particular data structure we have. Even just with a simple linear, we basically capture almost all of the variance. So we're almost hitting the maximum R squared in this case. And so from this last data set, we see that strength and toughness can be modeled more accurately than stiffness and strain to break with these different regression models. And as I called out, the categorical labeling is much, much better for these data sets, indicating that we can't just give a composition and hope that that will be generalizable, which unfortunately was the goal at the beginning. So more work to be done there. So this summarizes, I think, a lot of the key findings across these three different data sets. So I would say mixed success. There are some properties that we can actually predict pretty well, others not so much. And there is moderate sensitivity to the way that we actually structure and clean our data. So again, if we only select whole beams, that's actually a really great way for us to improve our model accuracy. Imputing those values for the beams we couldn't test also improves our accuracy. And again, this point on categorical labeling seems to be really important for us actually getting models that perform well on this data. One thing that I think is interesting looking here is in our second data set, we see that the stiffness or this E subscript F for the biomatter analogs, that was the only property that we could actually model with above 0.8 R squared. And something that I thought I might find is that there would be one particular property that might be really easy to model and others might have more noise or might be more challenging. But that seems to actually not be the case when we look across these three. So while we do a decent job at the stiffness for the first two data sets, in the third, it's actually really, really poorly predicted. On the other hand, we see that our worst performance in the second data set is predicting the toughness or the integral of our curve, but it's the one that we predict the best in our last data set. And so this is, I would say, an open question to understand why do we actually see these different trends along what can and cannot be modeled. So that's most of the work that I did. I'll close with a couple of thoughts on things that I think would be interesting for us to look into. So the first one on the left here seeks to aim a big question of how can we leverage similar methods in data science to understand the best way that we can actually make our experimental design to maximize the ability to model a particular system. And so, for example, we can ask, is it better to uniformly sample across our processing conditions, or would we rather have sparse sampling with replicates? So that's kind of what we see on the left. Would I rather have a single sample, no replicates, but try more temperature pressure pairs? Or is it important for us to actually have replicates given the noise inherent to our system? In a more general case, we can ask, if I have n samples I'm going to make, which n samples should I make in order to best maximize my experimental efforts in order to understand how to map our processing or our composition onto properties and performance? And the second one, I think I pointed this out in the beginning. Not every stress strain curve is a triangle. In fact, most materials around you are not going to have a stress strain curve like this, but it is indicative of brittle materials like ours. And basically, it means you have a linear response between the force you apply and the deformation. Um, and a question that I have not answered, but I think would be interesting to explore is there any way to leverage the geometric assumptions or the geometric relationships here? So for example, we saw in our third data set, we can predict 2 out of these 4 properties really well. And if you think about a triangle and the area of a triangle, it's 1/2 base times height. So theoretically, if I know the strain and the strength with reasonable confidence, should I not be able to just infer that my toughness should roughly be 1/2 strain to break times strength of my samples, for example? Does that give us better performance than the models that I'm trying to, and in many cases, failing to properly create? Um, Open question, I don't know the answer. So those are some of the next steps. And with that, I'm going to go ahead and thank the Roumeli Lab, particularly Professor Roumeli, but also Ian Campbell and Harish for doing the actual work to make the samples, as I said. And then I also want to call out Owen Parks, who is an undergrad who has been working on this project with me. And with that, I will invite any questions about what I talked about. Thank you very much.